All right, everyone, welcome to Unit 3. We are starting the urinary system, and remember the first half of urinary system was self-study. There was an assignment and a reading assignment that went along with that. Not to mention, if you haven't already done that, then this lecture over the second half of Chapter 23 isn't really going to make a whole lot of sense because I'm going to refer to things that were in that self-study part. So if you haven't already done the first half of this chapter, I would advise waiting to watch this video, going back and doing that self-study portion, and then coming back and picking up with this video. But let's go ahead with our not really attendance questions, and I'm going to do it a little bit differently today. In the past, I have used a pop quiz when we're doing our in-person classes, and that pop quiz is over a lot of material from that first half of Chapter 23. So let me pull that up. And again, this quiz is nothing to do with this class. This is from an old pop quiz that I used to give. And let's see what these answers are. And really, before I go over these answers, you should try to go over them and answer them yourself, just like you would with all of our other questions that we use for not really attendance. Number 1, what hormone is produced by the urinary system when oxygen levels drop in the blood? Think back to when we were first covering blood. Erythropoietin, EPO. When the urinary system plays a role, the urinary system plays a role in the activation of which vitamin? Vitamin D. This ties in with the skin chapter from Bio 137. Remember, there's cholesterol that's moved up into your skin, and when ultraviolet rays from the sun hit that, it's converted to an inactive form of vitamin D, and then that vitamin D inactive form is moved to your kidneys where they activate it. And vitamin D, recall, is needed to absorb calcium from our diet, which we talked about in the bone chapter in Bio 137. So that is why they say that going out in the sun is good for your overall health because of vitamin D. What are renal columns and what are renal pyramids? Remember, when looking at the structure of the kidneys, the outer portion is called the renal cortex, and the inner portion is called the renal medulla. And that renal medulla is actually made up of these small triangle structures called renal pyramids. So each kidney has roughly 8 renal pyramids, and collectively they make up the renal medulla. But in between those pyramids, some of the renal cortex extends down, kind of separating one pyramid from the next. And that cortex portion that extends downward, those are called renal columns. List the organs of the urinary system. Well, we've got left and right kidneys. We've got left and right ureters. We've got a urinary bladder, and you must specify urinary bladder. You can't just call it bladder. Remember, there's more than one bladder in the body. And there is the urethra. Those are the organs of the urinary system. What does retroperitoneal mean? Well, retro means behind, and peritoneal refers to the peritoneum. Remember, from the very, very beginning of Bio 137, the abdominopelvic cavity is lined with that serous membrane, that 2-layer membrane called the peritoneum. Well, the peritoneum wraps most of the organs in the abdominopelvic cavity, but behind the peritoneum is where we find the kidneys, and that's what retroperitoneal means. The right kidney is displaced by the liver and lies slightly lower than the left kidney. The renal plexus is primarily which branch of the nervous system? It is primarily the sympathetic nervous system, the SNS. What is renal ptosis? Renal ptosis, that is when a kidney drops and sits lower in the body than it should. It's called a dropped kidney. And this is dangerous because it puts stress on the ureter, and it can actually kink the ureter and cause a backup so that urine can't leave the kidney. What is a nephron? A nephron is the functional unit of the kidney. And that's what this lecture, these next several lectures, are really going to focus on, is how nephrons work. Approximately how many nephrons are in each kidney? Over 1 million. I believe our book is a little more specific, and it says, I think, 1.2 million per kidney, but it's over 1 million per kidney. And the two major components of each nephron are the renal corpuscle and the renal tubules. Now, the renal corpuscle we're going to look at in these lectures. The renal corpuscle is actually composed of two structures, the glomerulus, which is a modified capillary, and Bowman's capsule, which is kind of a shell that surrounds the glomerulus. And then the renal tubules will have different segments, which we will really focus on in these next few lectures. All right, so let's move back to our lecture PowerPoint. Again, it's going to be very brief. It's just going to be an introduction to how the kidneys function and the first step of urine formation. Now, when we think of the kidneys, the first thing that we think of is those are the organs that make urine. But as I alluded to, on the first day of class, the kidneys are there to cleanse your blood. So there's that tie-in to blood that I said every chapter this semester is going to have. The kidneys are what cleanse your blood of things that our body produces as waste. Now, also, something else that I mentioned when we were talking about blood pressure, I said don't flush that blood pressure information out of your head, because really once we get to kidneys, that is going to be blood pressure part 2. So in addition to cleansing your blood, your kidneys also will help maintain blood pressure, and they do that by maintaining blood volume. We talked about that a little bit back in cardiovascular. And something to tie into the chapter that we have coming up after this, your kidneys also can help adjust and maintain your body's pH. So your kidneys do a lot more than just normal pee. So let's talk about how kidneys actually make urine. And while that is what we talk about all of this entire chapter, it's all going to be talking about how kidneys form urine. Think about why we're forming urine. It's not just to get rid of waste. Now, when we talk about urine formation, there are a few steps. We're going to talk about 4 main steps of urine formation, and these terms will actually mean something very, very specific in this chapter, and it might be a little bit different from what you're used to hearing these terms mean in other settings. So the first step of urine formation is filtration. So filtration is where your kidneys actually remove substances from your blood and place it into the nephron. Now, this occurs at a specific part of the nephron. It's inside Bowman's capsule at a special capillary called the glomerulus. So over here on the right, we see a very generalized nephron. Here we see the blood coming in, going through this weird looking tangle of blood vessels here. This is a modified capillary called the glomerulus. This is where filtration takes place. Filtration, we are taking substances out of the blood, placing it into the filtrate inside Bowman's capsule. So that's going to be the definition that we go with. Taking substances out of the blood, placing it into the filtrate inside Bowman's capsule. Now, that brings up this question, filtrate. What is filtrate? Filtrate is very similar to plasma. There's going to be some differences. We'll see those later, but it is almost exactly the same thing as plasma. It does not have the proteins that plasma has, and some of the ion distribution will be a little bit different, but it's very similar to plasma, and by the time we get to the end of the nephron, it will be considered urine. So filtrate is almost plasma on its way to becoming urine. Now, once we have done this process of filtration inside Bowman's capsule, that filtrate is now going to move through the rest of the nephron, called the kidney tubules. And as we move through the tubules, we're going to take that filtrate, we're going to take some stuff back out of it because our body might still need it. And we're going to put some more stuff into it that wasn't filtered because our body still needs to get rid of it. And those two processes are called reabsorption. Reabsorption is taking substances from the filtrate and moving it back into the blood. This happens all along the entire nephron, taking substances out of the filtrate and moving them back into the blood. And what kind of things are we reabsorbing? Well, 100% of the glucose that is filtered is reabsorbed. 100% of the amino acids that are filtered are reabsorbed. 100% of most vitamins that are filtered are reabsorbed. And 99% of the water that was filtered is reabsorbed. So really, practically everything that was filtered is ultimately placed right back into the blood because our body still needs it. Now, even though it's, in my opinion, really considered a part of reabsorption, we also, in our book, see that it says reabsorption and water conservation. Really, the water conservation is a part of reabsorption, so I will just refer to reabsorption, which includes the water conservation part. Now, some things might not have been filtered up here for various reasons we'll see later, and our body still needs to get rid of them. And that's what secretion is. Secretion is moving substances from the blood into the filtrate along the kidney tubules. Moving substances from the blood into the filtrate along the kidney tubules. And then the last step, excretion, this is what we do at the toilet. This is removing the urine, the finished product, from the body. So these words all mean something very, very specific. Filtration, reabsorption, secretion, and excretion. Now, we're going to see this drawing here quite a bit, and this is a very generalized nephron. Don't worry about all the stuff that's written on it for right now. Pay more attention to the shape. Books will always draw nephrons that look kind of like this. Here we see this red. These are the arterioles and the glomerulus. Here we see Bowman's capsule, the first part of the nephron. And then we see the proximal convoluted tubule going across, the descending limb of the loop of Henle, the ascending limb of the loop of Henle, the distal convoluted tubule, and the collecting duct. Now, I will use abbreviations when I write these out. I will call this the PCT, the DLOH, the ALOH, the DCT, and the CD. And the reason that your book and other sources will show a nephron like this is because it's really, really easy to indicate. Right here, I'm talking about the PCT, and I can make notes. This is what happens at the PCT, and it's easy to indicate it. But even though this is the easy way to draw and talk about a nephron, I want you to keep in mind that this is more what a nephron actually looks like. And it'll be important to keep in mind this shape a little bit later on in a future lecture when we talk about some of the activities of a nephron. It's important that we understand exactly the layout of the nephron. So we still have the arterioles, the glomerulus, Bowman's capsule, the PCT, the descending and ascending loop of Henle, and the DCT connected to a collecting duct. But it's just, we couldn't really easily indicate everything if we drew it the way that it really looks like. Not to mention, I'm not an artist. I could not draw this if I had to. So just know that nephrons are drawn the way that they are for a reason. And something else that we see in this picture that'll be important later is this kidney tubule is surrounded by lots and lots of blood vessels, mostly different types of modified capillaries, which is important to think about. If we're reabsorbing or secreting something, we need those capillaries to lay very, very close to the kidney tubules to make it more efficient. But we'll come back to these pictures a little bit later. Back to urine formation. When we're talking about forming urine, there's about 180 liters per day of plasma, of blood, that is filtered. So that blood that passes through the glomerulus, about 180 liters of liquid is removed from that. And that's about 47 gallons. And that 47 gallons will move through those kidney tubules. Most of it, again, is reabsorbed. Less than 1% of that leaves as urine. So the other greater than 99% is ultimately reabsorbed as it passes through those tubules that make up the nephron. And it goes right back into circulation. Now the kidneys, because they are doing so much processing, they require a lot of ATP, which means they need a lot of oxygen. Your kidneys use anywhere from a fifth to a quarter, 20 to 25% of the oxygen that your body needs is going to be used by your kidneys. So now let's look at the glomerulus inside of Bowman's capsule. And again, remember the glomerulus is just a very modified capillary that came from the self-study part. And what we see here in this picture, this is the capillary. This is the glomerulus. Over here is inside Bowman's capsule. So if we're filtering substances, it's moving from the blood into Bowman's capsule. And we say that it is crossing the filtration membrane. Now, what is the filtration membrane? It is the wall of the capillary. Remember, capillaries are a single layer, simple squamous epithelium. And the podocytes, which we'll see in just a moment. These are special cells that surround the cells that make up the glomerulus. And then we have this membrane here in between them made up of special proteins and carbohydrates. So when we're talking about filtration, this is a passive process. It doesn't require any ATP at all. It's actually all driven by pressure. Pressure is going to be a big topic of our next lecture. And when we talk about the glomerulus, it is fenestrated. Think back to when we talked about capillaries. Fenestrations are basically holes in a capillary, which is what we see right here. The adjacent cells that make up the capillary will actually have some gaps in between them. And these gaps, these pores in between the endothelial cells, the cells that make up the capillary walls, the pores are about 70 to 90 nanometers wide. So substances can actually exit through those pores but then they have to cross this part of the basement membrane called, made of these things called proteoglycans. Now, these are proteins and carbohydrate chains that makes this jelly-like substance. It makes it hard for stuff to cross. So now we're going to start to be selective with what can cross this membrane. Some things are too big to exit through the pores. Some things that can exit through the pores are too big to pass through these proteoglycans. The proteoglycans block anything that is bigger than 8 nanometers. And even things that are 8 nanometers or smaller are repelled if they are anions. Remember, anions, think AN, a negative ion. So anything with a negative charge is also repelled by these proteoglycans. So let's see, what cannot get filtered out of the blood here? Blood cells, they're much too large. They are way too big to exit through these pores. Plasma proteins, again, way too big to leave through these pores. Large anions, things that are bound to proteins, which are certain drugs, certain minerals. We'll see those a little bit later. Since they're bound to proteins, well, the proteins can't leave, so neither can the things bound to them. Anything bigger than 8 nanometers in diameter, too big to fit through. And then again, if they are anions, they can't get through. Now, over here, what we see, these little projections right here, these are what we call foot processes of podocytes. Foot processes of podocytes. Let's go over here and look at this kind of cartoonized image. This red tube is the capillary. It's in this case, it's the capillaries that make up the glomerulus. And then this big yellow thing, this is a podocyte. This is the cell that surrounds those capillaries. And the podocytes have these long projections that wrap around the capillary wall, and then these smaller little projections that extend from that. So it's like arms reaching out with some fingers spread out. And remember, the capillary, the glomerulus, is fenestrated. It's got these holes. But these foot processes can actually cover or uncover these holes to allow more or less to be filtered. So these foot processes, these pedicels, will allow or prevent things from being filtered. So what can or can't be filtered? We've mentioned some of them already. Things that are very small can be freely filtered. If they are less than 3 nanometers, they will pass right through, and they'll be found in the same concentration in the plasma and in the filtrate. Anions, negative ions, are not filtered. Regardless of size, they are not filtered. And small protein-bound molecules are not filtered. Again, regardless of size, they're bound to proteins, and the proteins are much too large to be filtered. And that's things like calcium, iron, thyroid hormones, lots of drugs that are carried in your blood, not necessarily illicit drugs, but things like, for example, something is bound, maybe penicillin. Penicillin is bound to a plasma protein. So over here we see things that do pass through: water, electrolytes with a positive charge, glucose, amino acids, fatty acids, vitamins, urea, uric acid, creatinine. Those things will freely pass through. So here is kind of a look at the glomerulus. Think back to when you were doing the self-study part. We have the afferent arteriole bringing blood in. It passes through this very modified capillary, the glomerulus. Lots of substances are filtered into Bowman's capsule. Anything that is not filtered then leaves the glomerulus through the efferent arteriole. Now this glomerulus has had Bowman's capsule and all of the covering stripped away so that you could see it. Over here on the right, we can see this is one of those podocytes, and it has all these foot processes extending completely surrounding the glomerulus. And down here, magnified even further, this is that filtration membrane. Here's a cartoonized version showing the efferent arteriole coming in. Here's a cutaway view to see inside the glomerulus. Here we can see those podocytes covering it. Anything that's not filtered leaves through the efferent arteriole. Anything that is filtered collects here in Bowman's capsule, and as Bowman's capsule fills up, that filtrate leaves through the proximal convoluted tubule. Over here on the right, we see that same thing through a microscope slide, and this is one of the slides that you will need to know for lab, which you can find on my YouTube channel as part of the Exam 2 slide review. Now, let's look at something that can go wrong with the kidneys. We'll talk about some signs of kidney damage. You might find protein in the urine. You might find blood in the urine. You might find a few other things in the urine. And something from the lab you may have learned if you looked at Lab 8 yet, there's kind of a giveaway when you see something in the urine. If you just add the word urea to the end of that word, most of the time that's what you actually call it. So when you find protein in the urine, that's called proteinuria. That's a sign of kidney damage because you shouldn't find protein in the urine since it wasn't filtered. Hema, remember hema meant blood, so hematuria is blood in the urine. And again, we shouldn't find blood in the urine because the blood cells are too large to be filtered. So proteinuria, protein in the blood, proteinuria, protein in the urine, I apologize, and hematuria, blood in the urine. And those things are signs of kidney damage. Now, this is where I'm going to end for the day because tomorrow or the next day when I post the next video, we're going to actually start talking about the processes that occur that actually drive this filtration. And what I would like for you guys to do before you watch the second video in this setup that we have now, I would like for you to go back, way back when we were talking about capillary fluid exchange at the end of chapter 18, I think it was, or 19, whatever chapter the blood vessels were. Go back and review that capillary fluid exchange because that's going to be exactly what we talk about next time. Those same exact pressures, but with different values, are going to be what drives filtration. Because again, remember, the glomerulus is a capillary. So those pressures inside and across the capillary are going to be what actually filters our blood once we get to the glomerulus. All right, so it was a brief lecture. I'm sorry if the background noise was distracting. Don't hesitate to ask questions if you have them while you're watching these. Text me, email me, let me clear these things up for you because I know getting them delivered this way is going to be really, really challenging for some of you. So let me do everything that I can to help this all make sense. All right, I will record another lecture video tomorrow or the next day and post that just as quick as I can. Keep an eye on YouTube for those lab videos. There's several of them already up. and I will be in touch. Everybody stay safe.